think we're good to go. So we started talking about this last time. Let me recap a little bit. So the global picture is we're talking about hidden messages. Okay, I talked a lot about the biology background. Let's not recap that as well. The specific hidden message, we're looking at our genes, and because of the genetic code, there's some hope that you can detect these hidden messages because there's a specific start codon, a 3-letter subword that marks the start of a gene or could potentially mark the start of a gene. And then there's 3 options for the stop codons that kind of has to mark the end of a gene. If there is a gene, then it has to eventually stop. And by and large, it has to use one of those 3. I'm sure there's some exceptions in some weird cases, but this by and large seems very stable. Now, the question is this good enough to detect genes? And the answer in principle was probably yes, because most genes that humans have, or I think this is more generally true, they're fairly long compared to what you would expect if you just generate randomly mutated DNA and look for the longest stretch you can have between such an open codon and one of the stop codons. We call that the open reading frame. So it's a long substring that does not contain any of the stop codons. And if DNA were random, or if there was no reason for evolution to stop mutating away from a specific place, then it would do so. That's the assumption. And then we would see only very short such regions, such open reading frames. So in turn, if you see in some actual genome a very long open reading frame, it's probably an actual gene. You're looking at this in the tutorials a bit more on exactly how to compute this null model. Don't want to revisit that in full detail. And I want to come back to this model briefly. So this was the description of a language of strings that does not contain any of the stop codons except for as the final trailing occurrence. So it's all strings that have no occurrence of the stop codon except for a single one, which is the last thing in that string. That we could describe as a finite automaton. And from there, using essentially Markov model techniques, you could compute the expected length of a sequence of steps through this thing, if each step is taken uniformly at random over the letters, assuming the null model of DNA. It was relatively easy to get the expected time, and what we did is essentially locally write down, have a variable for the expected number of steps starting at a specific state, and then we could express in a sequence of equations how these relate. If I'm at this accepting state, then the expected further steps is 0, and for all the others, there's some random component that tells me where to go. I'm always adding 1 step. That's happening for sure. And then depending on the random outcome, I might have more steps to take. That gives me a system of linear equations. And okay, I'm not going to show it again. We solved this essentially using SymPy. Yeah.
What is automaton? What's the first input base for this automaton? Is it the one right after the start codon? Or is it the middle one of the start codon because these are overlapping?
I think here, if it was the middle one, then it would have to be U, right? I'm assuming we read the start codon and then we start. So okay. It's AUG, and then we jump into the start state.
So it's the fourth base.
Yeah, that's right. But then in the numbers we count here, it's not counted, right? The start codon is excluded. You can add 3 or not add 3. it's more about teaching the technique, which certainly is also more general than the specific example. Right? You can do this with any kind of strings that you want to avoid and count how long you avoid them in a random string. What I want to get back to is doing more than just the expected value, because that here is maybe even good enough, because the difference between what we observe as open reading frames and actual genomes and this expected value is so large that you can possibly stop there and say, it's qualitatively so big a difference, I'm not going to do real statistics. But suppose we want to do real statistics and calculate the probability that we see such a large reading frame under the null model. A nice general tool to get more information about the distribution are these probability generating functions. We defined them last time already, so I'm going to quickly over. If you have some random variable and it has non-negative integer values only, then you can write this kind of series. Could be infinite or could be finite, both works. And that's a formal power series, or you can also say it's a representation of a function in terms of its power series. And so that's a function in some variable set. We'll mostly treat it as a symbolic object, and that's where we left off. One convenient thing is to compute the expected value of such a variable. If you have the probability generating function in a convenient form, that's easier to do than from scratch, because you can just take the derivative with respect to set. Only one variable, so just the ordinary derivative, and then set to 1. And that's going to give you the expected value. And here's the proof. It's a very simple calculation. All you need to know is that the derivative of a sum is the sum of the derivatives. And that means we take the derivative inside the sum here of each of these terms. And that gives us precisely this formula. Once it's set to 1, that one just goes away. So if I was to insert 1 here instead of the general set after taking the derivative, then this goes away. So the whole thing is kind of just gone. If I strike it through, can you still see it? I think it's clear what happens. And then this is just the definition of the expected value if you keep the whole thing with the sum in front. So that's cute, but maybe not so exciting, because we knew how to get expected values even before this. The nice thing is it also works for other moments, and we'll look at the variance as an example. And there, the formula looks a bit more annoying to memorize, but maybe you don't even have to. The kind of trick that happens is the very same. If I take my derivative here, but now I take the second derivative of this inner part, just the summans. Well, I can't do this really in one shot, but we have the first ones here, so it's not too bad to do. That's just a constant factor if you look at it as a function of z. Same for the k. Now we have another polynomial. Essentially, that's going to be the same pattern. And I guess technically this is only true if k is greater or equal to. For the smaller ones, we don't need to worry, because then there's a factor of 0 in front and the whole thing is 0. Right? For those small powers of k, the factor in front will kill the thing. Okay. So if you sum up just this part, so again, if you set z to 1, this part goes away, and we have the probability to have value k, and then we have something that looks similar-ish to variance, but not quite what we would want for the variance. is, you can say it's the expected value of x minus its expectation squared. So we sum over all possible values for k. Say k greater equals 0 again. Probability for x to be k. And then this lovely thing. And we already know what this is, right? This is exactly this term. So that you can insert. And the easiest now is to move backwards. If we look at this term on the right, let's expand this one more step. Okay, I'm gonna write it again. And we have k squared minus k 2k ex minus ex squared. Then you can expand this one out. That goes completely out of the formula. This last amount doesn't depend on e of x. This is a constant, so it can go outside. So here we have the k squared. Here's the k. That's a plus. And here this is already easy, it's summing probabilities, so that's going to sum to 1. Okay, let me check. So this one's our expectation again, right? So we have 1 ex squared minus 2 ex squared. That leaves us with the 1 minus ex squared. And this one, we can split up again to look a bit more like this one. K squared is the same as k times k minus 1 plus 1 plus k. And that's where we have another e of x from. Okay. Maybe there's a more elegant way to do it without as much calculation. The nice thing about this taking derivatives of the probability generating function is it generalizes to higher moments without much problems. As long as you can take derivatives, the rest is just some rote calculation. We'll leave it with expectation and variance. Then you can use typical statistics on it, or you can use Chebyshev inequality to get bounds on large deviations, these kind of things. You look at that in the tutorials a bit. Let's also look at some example for a concrete distribution where maybe I can convince you better that it can be convenient to use these generating functions. So sorry, there's the view part on the slides missing it that I didn't copy. Good. We don't want to do this from scratch. I'm sorry. There was this question from last time that essentially will be answered in the tutorials. That's better. So while we're there, this depends a bit on your assumption. One or the other could be more sensible. And I think there's no definitive answer that says one or the other is more correct. It depends what you assume, whether you can reliably, with the start codon, detect the right position, or if you also have to live with the fact that it might be offset and you still want to define reading frames. So you look at both options. I did the one in lecture that gives the more interesting math. As you'll see in the tutorials, the other one is easier to deal with. Okay, with the slides that have the update that is not online yet, I don't have to write this example from scratch. I'll use this notation throughout. So uniform and then a set just means the uniform distribution over this discrete finite set. Here it's the first n integers, where obviously we start counting at 0. So every value with probability 1 over n. Then the probability generating function is just a polynomial. It's a finite sum of powers of z. And the common factor, the probability is always 1 over n. So I can write this like that. Now, that's just by definition. The power of this comes in because I can do algebraic reformulations of this term. In particular, just in case people might have forgotten about this, there's the TCS cheat sheet. And there's a formula for summing up powers from 0 to n. So that's exactly the form we need. And if you do that here, take the first n powers. So the formula on the sheet went until n, so I had an n plus 1 here. In our case, it's even more convenient. It stops exactly at n. So we can rewrite this sum in closed form. Okay. So it's shorter to type. And now we can take, for example, the derivative of this thing, set it to 1. Or I take the limit as set goes to 1 and retrieve, ta-da, the expected value of a uniform distribution. What is a little less easy to do in your head is the variance. I can never remember. I always have to look this one up. But if you're, say, stuck in a lecture room and would have to reconstruct it, that would be the one that I think is most foolproof, so that you can do it without making calculation errors. Okay, let me, just for the sake of the argument, show that you can do this in SymPy if you wanted. It's the same I started with before. That's this one. So just to show you that very little code suffices, is a little bit of a plug for Python here. There's this symbolic computation module in Python, and we import some functions that we're using here in the next few cells. You have to define symbols. That's what you're working with in the formula, but then you can write down terms in a very natural form. Like this generating function here, you just express it as a standard Python expression. And you can take derivatives and you can set things to 1 with the subs function that we've seen last time and is used here as well. Now, if you do this in this one, you get a 0 by 0 because you have this set minus 1. There's a root in the denominator and a root in the numerator, but they actually cancel if you look at it carefully. A simple way to do this is to compute limits, which SymPy also is, for these simple cases, is doing very well. I'm just showing this also to educate you about tools that are convenient to use. And you can take derivatives twice, add the expectation, subtract the square of the expectation. That's just from this variable here. And then you get the variance of the uniform distribution of the first n integers. Okay? I find this cool. I don't know if enthusiasm sprang over. There's one more cool thing that I wanted to show. This is, if you have the probability generating function of some random variables, you can, and they're independent, stochastically independent, you can get the generating function of the sum of the two very simply as the product of the generating functions. If you write this down, this is nothing but the convolution. So what does it mean to multiply products of this form? Let's take the symbolic general form. I have two generating functions. They'll be of this form for some sequences, AK, BK. The product is also a series. And at the kth term, it has a sum of all the options how I can build K. I mean, think about this as the finite case, two polynomials. You multiply every combination of terms, and you combine all of the terms where the resulting power is exactly K. Right? So you can have from the first series one contribution set to the L, then you get AL, and you multiply that by BK minus L. And that's one of the terms that you're summing up for the resulting sum where the power of set is K. In a way, this is just the definition of how you multiply power series anyways. It follows from the usual laws of how we do this for finite cases. And it works just the same if the sums are infinite series. And for the probability generating functions, what you get here, AL and BK minus L, that's the probability that X is equal to L. That's the probability that Y is equal to K minus L. If they're independent, then the probability that they jointly have these two values factors, and then you get exactly that this is the probability for X to be L and Y to be K minus L. Of course, then their sum is K. And there's you sum up over all different options for the value of X, say. Okay. Simple. That formula is also somewhere on the cheat sheet, if you can't remember it. Let's do a quick example here as well. This one I also find very useful to memorize stuff. Say you have Bernoulli random variables, so just coin flips with a bias, so they come up with probability P to be 1 and 0 otherwise. Then the probability generating function of this simple coin flip is very easy. P times Z for the one case and the other case. Now, standard binomial distributed variables are the sum of n of these things. And that means independent Bernoulli trials, I should stress. This is not on the slide anywhere. Okay. I guess all need to be mutually independent. But standard stuff, I'm not telling you much new. The probability generating function of this binomial random variable, though, is then the product of all these individual ones. The product of these is all the same, so it's just the nth power of that generating function. Okay. I rewrote this here so that it's clearer how you take derivatives, because that's, of course, what we want to do next. If you want to compute the expected value of y, I know that's a bit silly. You just want to go via the definition, but you could just take the derivative in this guy and set to 1. So what you get is the inner derivative is p, and the outer derivative is the n, the power goes down, and then the inner with power n minus 1. If you set to 1 in this one, you notice the p here and the minus p always cancel, and you get a 1 to some power, which just goes away. So n times p, as is obvious from the stochastic representation, the expected value of one of the xi's is p, and we got the sum of n independent copies. Okay, that's not hard to do otherwise, but the variance I can never quite remember, and you don't have to. You can take the formula from before. If you take the second derivative and plug in, then you get this lovely term, and the other two are the expectation minus expectation squared. And you can rewrite this in the somewhat more typical form, n times p times q, when you set q to 1 minus p. That's the one I learned in school. And, yeah, maybe in school, I don't know. Also here, very briefly, like it's a proof of concept. There's the calculation, and you can even use simplify to do the combination of terms I showed you. SimPy immediately gives this representation as the shortest. Okay. That's all on the generating functions. That directly connects, that directly like goes from the definition. I promised that we come back to this automaton. So let me take a copy of this one. Doesn't work. We can apply the same approach that we did for the expectation with the full distribution. If we say that our x and then the state, say p for prefix, that is now the generating function of the number of steps from this state till you reach an absorbing state. Let me write that down. It's the probability generating function of the number of steps x is a strange letter. Let's say this is the number of steps as the random variable to some absorbing state. And that's a random variable now. And we could say gp of z as the generating function. Of this guy. And then you can do the same kind of reasoning as before. Let me do an example. So the GUAA, which is an absorbing state, there's no further steps to be taken. So it's the deterministic constant 0, which means the probability generating function is just a 1, z to the 0. And let's do the UA, for example. Maybe this doesn't need to be quite as small. If you look at the UA, there's some chance with probability 1/2 it goes to one of these absorbing states, but let's write it down explicitly. So we do one more step. There was a +1 in the original formulation. Now we have to treat this as the sum of two independent random variables, one of which is the deterministic constant 1, and the other is as many steps as I'm taking from the target state. So the constant 1 has a generating function of just z to the power 1. The product of the probability generating functions corresponds to the sum, so I'm multiplying. And here I'm going to just take GUAA That's what I get in this first case. Then I get symmetrically for the second UAG. Now I know it's silly, these are just one, I could have inserted them straight away. But here, this is showing the general pattern. If you go with a U back to this place, then you get the same pattern and just there's a GU now instead of this one. And the last term goes back to the start state. And you see how you can read this off from the automaton without thinking, like the first part here is even the same all the time. Okay. So you can equally form a system of equations. Don't be scared because there's a variable set flying around. It's not biting. You can just symbolically solve the equation the same way. And you'll get some solution for the G epsilon as a function of Z. All the others can be eliminated because you have as many equations as variables. So you can determine the exact representation for all of the states, but we're interested in the one from the start state. Again, this I prepared. So here's the full list of equations. Is again written in this SymPy convention where an equation has to be rewritten so that the right-hand side is 0, and then you just give the left-hand side. That's the start state, and I'm subtracting, well, there's 1/4 chance of going to U, and there's 3/4 of a chance of staying in the start state. And you can check in the automaton that that's what happened. Let's do the UA one was the one we did. You really see how these are very irregular and they're essentially just giving the automaton edges. If you solve that, that's the same code as last time, so I'm not going to explain the SymPy specifics again. There's a solve function where you give equations and variables, and then you can get the solution is a map from the symbols to the solutions. What we care about is the one for t. So it's t as a function of z. You can set to 1. What should be the result of this one? Double checking if people are here. That's the solution for the probability generating function of the start state. So the number of steps we take from the start state. But I'm just inserting z equals to 1. So that thing will just sum up all the probabilities. So it has to be 1. If I substitute 0, I guess I get the probability that it takes 0 steps. It should be 0 in this case, because from the start state, we have to take at least 1 step. Then we can take derivatives and insert 1 in the derivative. That gives us the expected value. That's what we already know, but we should double check. And then we can take the second derivative and compute the variance. I don't know why this is underlined. I think the code runs fine. This is the generating function. The way it's determined, it's from a set of linear equations, so it's always going to be a rational function. This is maybe not so obvious, but you can prove this is always leading to a function polynomial divided by another polynomial. And these are very tame to work with, but we're not going to dig deeper into this here. So t of 1 was 1, summing up all the probabilities. Probability to use 0 steps was 0, as we said. That's the expected value, same as we had last time, if you look it up. And the variance is, well, whatever, some number. But more intuitive, I think, is the standard deviation. And so if you do this cowboy statistics of saying expected value plus minus 1 standard deviation is a reasonable confidence interval, then you're still far away, like you're still within a couple of dozen bases in an open reading frame of a random DNA, far away from the length of actual genes, which was usually thousands of bases. Okay? Good, that's all of my detour to generating functions. We'll go on to something entirely different, which may or may not help people. We'll see. This would be a natural place to do a little break because it's in the middle of the topic. Or shall we keep going? There's a video. We can do a break after the video. We looked at one type of hidden messages, which was just a gene, a region in the DNA that codes for a protein. This connected to that, I guess the biological backstory is a little more subtle, but is, in computer sciencey terms, a stronger and more easy to handle hidden message. And that's motives. I'm going to explain where this comes from, what this is. For that, a little detour on why it's interesting, and that is a biological motivation. I talked about genes being portions that code for a protein, and then we talked about this process, the central dogma of molecular biology, that the genes are translated to transfer RNA, which then in the ribosomes is sequenced into proteins. And that's how we make all these chemicals that fulfill biological functions. Now, if that was the only thing, every cell that has the same DNA would have to produce the same proteins all the time. And it wouldn't really be possible to have the same genetic information and yet have different cells do different things. And so there must be something else going on. And much of what's going on is called gene regulation, which is, I think the picture, okay, the video is Amoeba Sisters again with their particular style, but the content I think is at the right level and also accurate, and the drawings are a bit whimsical. It's a switch that can turn genes on and off. The way this works is going to be explained in a bit more detail in the video, but the kind of short version is the translation of a gene producing a copy of transfer RNA from the gene needs to start somewhere. And there's a special molecule, the RNA polymerase, that binds to a certain starting part of the gene and then starts the copying. And if you block that kind of electromagnetic starting position by putting something else there, another molecule, then that translation is not happening. Or if you make that starting region more attractive by helping it attract polymerase with other molecules, you can actually generate more of the protein. And that's what this gene regulation in the simplest case is about. Let's do the quick video here. And I'm muting my...
When I was a little kid, I had this art teacher. She had poetry, which I love. I got it on someday. And let it out. But that's creative expression. Our cells have expression
I'm skipping
too.
the poetry
Gene expression.
motivation.
They can express genes. To express a gene means that a gene can be used to make something functional, often a protein. In our protein synthesis video, we talk about these steps. The gene, which is made up of DNA, can be transcribed into mRNA during transcription, and then used in translation to make a polypeptide chain. A protein is made up of one or more of those chains. But you know what? It's not doing that with every single gene. Not every gene is expressed. That's why the phrase gene regulation gets put with gene expression, because the expression has to be regulated. A cell in the eye has no need for using a gene that codes for stomach acid, even though the gene is present. It would be wasteful to express that gene and likely uncomfortable. Prokaryotic cells and eukaryotic cells both have genes to express. Some differences, though. Recall that prokaryotic cells just have the DNA in their cytoplasm. They don't have a nucleus. So transcription and translation all just happen here. Unlike in a eukaryotic cell, there's no traveling of the DNA out of a nucleus. Nope, in prokaryotes it's all right there together and can really both be happening together at the same time as there's no nucleus involved. For prokaryotes, a lot of the control of whether a gene is going to get expressed or not tends to involve or impact transcription. Eukaryotes also have gene regulation that involves transcription, although they'll have a lot of other times for gene regulation too that we'll get into soon. So you might wonder, how does gene regulation involve or impact transcription? Transcription, again, is when an enzyme called RNA polymerase makes mRNA from a DNA template. There are regulatory proteins that can decrease or increase transcription, and that's important among both prokaryotes and eukaryotes. These regulatory proteins are often referred to as transcription factors. Some transcription factors bind to a DNA region called the promoter to help RNA polymerase start transcription, while other transcription factors can bind there to repress it. Some types of transcription factors can bind to enhancer sequences, where they increase transcription, and enhancer sequences can even be far away from the gene. Some transcription factors or other proteins can bend the DNA to bring the promoter and enhancer closer. Ultimately, transcription factors play huge roles in whether a gene is expressed, and we also want to mention environmental factors can influence transcription factors, meaning that the presence or absence of an environmental factor could impact gene expression. Prokaryotes commonly have operons, which feature a regulated cluster of genes along with a promoter and an operator, and they make great examples of gene regulation during transcription. We have an example of this in our Order of the Operon video, but here's a quick recap of just one type of operon known as a lac operon. Here's a repressor. The repressor blocks RNA polymerase from doing transcription, and it does this by binding to a sequence called the operator. Since the RNA polymerase can't work, ultimately these genes are not going to get expressed. That's because the genes are not going to be transcribed into mRNA, so it will not go to translation for a protein to be built. And that's great when the protein isn't needed. This is how the bacterium is regulating the genes in this operon. But sometimes the bacterium may need these genes expressed, and that's because these genes code for specific proteins, including an enzyme that breaks down a sugar called lactose. So when lactose is around, the bacterium wants that enzyme because it wants to break the sugar lactose down. It can eat the broken down lactose. So how does the bacterium turn a gene on? How does the gene get expressed? Well, as you can imagine, this repressor has got to be moved. It's in the way of transcription happening. And it turns out a form of lactose, technically an isomer of it, binds to the repressor, which makes it get out of the way. When the repressor is not blocking this RNA polymerase, transcription can happen. The RNA polymerase does transcription, and it makes the mRNA, and translation can follow, and it uses that mRNA to make protein, which includes an enzyme that can break down lactose, and other proteins too involved in using lactose. So these genes are being expressed when lactose is around. Expression is not happening when lactose is not around. It's a gene regulation example, controlling whether genes are expressed or not. And still on the subject of gene regulation that can impact transcription, we want to mention from our epigenetics video. Gene regulation that can impact transcription. We want to mention from our epigenetics video.
There's always a massive lag in the audio stoppings, but that's enough. I mean, as they alluded to, there's a few other things going on, and what the example they showed is basically for bacteria, for prokaryotic cells, where things are a bit easier. But the general principle is often the same. What do we make of this? A concrete example, which is from the book as well, why I brought it again, where they tell the story nicely, is the clock gene, if you want to follow the terminology there. How does it happen that living animals have a circadian rhythm, this roughly 24-hour cycle, that governs much of life? Now, of course, there's not a kind of universal constant that this is 24 hours. It's got to be the light of the sun that triggers this. But you usually wake up, if you have regular sleep cycles, you wake up before the sun gets up, roughly. And maybe even if not, then parts of your processes in your body start to get you ready for getting up way before the sun sets, before the sun rises. And so somehow there must be a clock started, and it's by and large started in the morning with the specific colors of light that you see in the sunrise. But then it kicks off a cascade of processes that have roughly this 24-hour duration. which can then be used to trigger things at the right time before the sun gets up again. And this wouldn't work if you were just instantly reacting to light, and then once the light's gone, you instantly get tired. Or, I mean, there's probably a lot more complicated things going on there. In animals, this seems to be more messy, but in plants, it seems there's a group of 3 proteins that have a fairly similar and consistent function in this regard. I'm not sure what these abbreviations stand for. I'm sure they have some long chemical name. I'm just going to use the abbreviations. There's one that, if it's present, it means there's more translation. TOC1 promotes the expression of these other 2, so more of them get made. With this kind of think about taking the block away, as in the video. Also, sunlight seems to trigger their expression. But then if these 2 are building up over time, and by the way, this protein synthesis, depending on how big the gene is not super fast. It might take minutes or hours to make some proteins, so this already adds a delay. And if these start building up, they themselves repress the expression of this other protein. So you have kind of a negative feedback cycle, so that over the day, a lot of these are made. They then repress more production of this TOC1 protein, which triggers more protein production of these 2. But once the sunlight's gone, these start to slowly get less. That means this TOC1 is no longer blocked from the high concentration of these 2 guys and can accumulate overnight. And that kind of sets a timer for when the night is over. If enough of TOC1 is there, it starts promoting the production of these 2 again, which is then enforced again by the light. But also before then, they start waking you up in well, whatever that means for a plant. I don't know. That's roughly the story. Now, the biology behind this is super exciting. But what this means for our bioinformatics question, there must be, plants do a lot of things governed by this circadian cycle. For all of those genes that react to the day and night cycle, there must be some way how these proteins influence their expression. And that can only really work if ahead of the genes, a bit upstream from the genes, there's a conserved pattern that these proteins can bind to stop or promote them being expressed. So we should be able in plants to find for many genes some similar sequences just upstream of where the gene starts. And that could be used in different ways. You can better understand how plants regulate day-night. Maybe you can identify new genes, and maybe you can tamper with that. That part we were not going to get in. And these kind of conserved sequences upstream of the actual gene, these are called motifs, or motif in general means, in this context, some shared common substring that's fairly preserved. As always in bioinformatics, don't expect the exact same string to be repeated in front of each of these genes. We'll have to live with certain little errors, mutations, and maybe also small changes that don't actually change the function, because there might be sort of equivalence classes of strings that work equally well. So the formalization that I want to look at here in the literature is known as the consensus pattern problem. I'll show you in a sec why that is a natural name. Ignore that name for the moment. Think of it as the motive finding problem. What we're given is a collection of strings. I call them G for genome, but really they're portions from the genome, which is the region just before the gene starts. And let's assume from the last chapter we know how to identify genes, so we can know, let's just read the 200 characters before the gene starts, and that's our G1 to GT. And we'll also give a length for this motif that we expect to find. And this again from the proteins that people know in plants are responsible for this. You can get some estimate. I think here you should think of K of being maybe 15 or a few dozen, not crazy large. Let's say or so. I mean, this is not a universal constant either. The goal is to find in each of these genes a starting position, so that if you read the next k characters from these positions, and you do this in all the different regions upstream of the genes. So let me do a small picture. Maybe the region you find here lies here, maybe here lies here. The positions are not really what we care about. What we care about is the content of these strings. If you write these strings on top of each other, so you should think of this being the first row here, this being the first and second row, third row, and so on, then there should be a fairly conserved pattern. And in formal terms, what we mean with this total Hamming distance, what we minimize here is the number of characters. So in each column, you determine what's the most frequent character, and you count the number of others. So here, this column, there's mostly T's. So 3 other letters give a contribution of 3 to the score, to the Hamming distance. And same for the other columns. It's a bit of it's maybe not the most natural generalization of Hamming distance, but that's the one that seems to be most natural for the biological problem. So if you have a column that's not changing at all, that's constant, then it's 0, and otherwise we just sum up all the columns. Is the problem clear? Okay, why is it called consensus pattern problem? Maybe you can already guess from this example. You can rewrite a bit the definition of this Hamming distance as the sum of distances in the normal Hamming distance sense from each of these red subsets, these substrings, to a specific string, one fixed string. And that's the consensus string that you build by taking the majority vote across columns. So here, because I took the picture from the book, there's other things in between that only come up later. The consensus string is this one down here. It just takes the majority letter or one majority letter of each column. And then the total score is the Hamming distance of every row with respect to this consensus string. And of course, you're just counting the lowercase letters first by column, now by rows. If you sum this up in the end, it's the same thing. All right, so the Hamming distance of the first row to the consensus would be 3, these 3 letters. And that G used to be counted here as a 1. That T and that T would contribute 1 to these counts. But if I'm summing up column-wise and then sum up those counts, or summing up row-wise and then summing up those counts, that's the same. So you could rephrase the problem here as saying we're looking for this consensus string. But it's equivalent. If you know the starting positions, you know which red strings we're looking at here, then the consensus string can be computed from that. And so that's the more specific formulation. You get the consensus string for free if you have those starting positions. Okay. I'll come back to the other parts later. The bad news first, the problem's hard, and even in a bad way. Sorry, that should be a T. It's NP hard even if the alphabet is small. They say even if it's a binary alphabet, maybe we would say the DNA alphabet with 4 letters is more natural, but that's not helping. And well, for the complexity theory people, even hoping for something that's a fixed parameter tractable algorithm in the number of strings is not going to work, very likely. No proof, but complexity theory assumptions aside, we shouldn't hope to find an exact solution to this problem in good time. Let's look at some simple things we could do. Brute force options to solve the problem exactly, just to see how bad things get. One option is we're looking for these starting positions in all the strings. I remember my picture with the red parts. I'm looking for these SI, S1, S2, and so on. So that if I read K letters on each side, that has the minimum distance to a consensus string. And I could just try all combinations. How bad can you possibly get? Well, that bad. You have roughly n options in each string, n minus k. But you have to have all possible combinations. It's not clear a priori how to separately optimize in each of the strings. And so both n and t should be expected to be large in practice. N is the length of the region that we're looking at. That's certainly a few hundred. And t is the number of genes, probably also in the hundreds. So that's not going to fly. What else could we do with the reformulation of the problem from here? Maybe we can first find the right consensus string and then worry about the rest. And that indeed also works. So you could try brute force over all possible strings for the consensus string. That brings the search space to sigma to the k, still exponential, but at least k is, if anything, that's the smallest parameter of the problem, because that's the length of the region we're looking for. And sigma, the alphabet size, is 4 in our case, so maybe this is, among the bad options, still the best. What do we have to do if we guess a consensus string? We still have to find these starting positions. But that turns out to be a lot more manageable, because if you're given a certain consensus string and you have one of those GI, it is independent of what the other strings choose. The best match between these two strings will be somewhere, and you can try all the positions and just find the one that has the least Hamming distance. And the key is that you can do this independently for the different genes. So that's a bit better. And overall, still exponential, but at least no longer in all the parameters. That means that this one here, I think, is for unbounded alphabets, if I remember correctly. Otherwise, it sounds like a contradiction to this. This one indeed is somewhat practical if your motive is small. What people in practice may do is use a smaller k, find a conserved region, and see if you can grow that region manually. But it's not really satisfactory. Like you're likely going to miss more important motives if you have to make k very small. Okay. For the specific example, this is not fast enough. Shall we still do a short break?
Could also stop 5 minutes early.
Good. Your call. Shall we have a vote? Stop early or do a short break now? Who's for stopping early? Okay, you win.
3 people working for you and 2 students.
Well. It is a bit concerning that almost half. Yeah, okay. There's some more people in the live stream who didn't get a chance to vote. Tough. That's the benefit of being here on site, I suppose. The rest of what I want to tell you maybe also is a short story. I want to present some maybe generalizable recipes for doing these heuristics. So solving a problem that's probably hard, where no obvious thing works, and we decide to give up the optimality guarantee. So we're willing to take some solution and essentially hope and pray that it's a good one, or check in with other means, and give up provable optimality. That happens more often than not in some bioinformatics problems. Now, yeah, in terms of heuristic, can mean many things to many people. In the context of algorithms, a heuristic is basically nothing. I mean, it's an algorithm that produces a valid solution to your problem. It's not gonna just give you some illegal output. But the guarantee there's nothing known about the guarantee of the quality of the output. It's to be contrasted to approximation, where an approximation algorithm has to have a specific guarantee. For example, C approximation means at most a factor C worse than the optimum. But heuristic, cynically you could say, is some person has one input where it worked, and that's as much as we promise. Yeah, if you had a bit more time, maybe it would be fun to brainstorm what you would do on these on this motive problem. You're given this collection of strings, and how do you even get started with doing something that's at least not taking forever, even if it might not find the best possible indices for the problem, the best consensus string. A simple thing you could try is to kind of go one string at a time and try to build the most sensible continuation of selections of strings. So for the first string, I'm going to go over all possible start positions. So I keep that brute force bit. Now if I fix some part in the first string, should have done that in red, I guess. Now I'm looking at the second string. How can I find something that is a sensible continuation of the red string? For that, let me borrow the picture from earlier again. One option, or one sensible option, would be to just say what we want is a region of that substring that doesn't have many mismatches with the first row. And maybe on a new page here. So you could say we're in this situation now that we've selected this string. And now we're looking at the second, we're trying to fill the second row, but we have a much longer string and we have to decide which one to take. Things are easy if we already have, as in this picture, highlighted which characters are eventually going to be to the contribution to the consensus string. And one option would be to say, I'm going to compute the consensus of what I have so far, and then find the best match for that consensus string. But it turns out that's a bit more blind than we can be, because it ignores things where there's a close tie between two characters, and then a third character is a mismatch to both, whereas one of the two characters might currently be in the minority, but might soon turn into a majority, and things like that. So what we'll do instead is look at the profiles as is explained, as it's called here. If you have, suppose this is our entire picture, so we've already selected a certain number of indices. We're further into this iteration, not just at position 2, but we're now adding the next row from the next string. How do we find that row? We compute the profile, which is we sum up in each column separately how often each of the 4 characters occurs. That's what the profile, or the, well, they call it count here in profile, and it's normalized to sum to 1. But of course, the same information. What that gives you is a way of saying, yeah, there's a column could be completely messed up in the sense that all characters occur equally often, or it could still be not a clear majority, but it's only messed up between 2 characters. Or it could be like this column, predominantly A, and you can quantify that. That's what's meant by profile. So you take your current selection from the starting indices you have, you take the current selection of k mirrors, of length k substrings. In each column, you separately count how often the letters occur, and you turn that into probabilities. I'll comment on the zeros in a second. So that's what's meant by profile here. Okay, that's this one. So there's a different profile for each position, and what the profile is a probability for each letter. Then I can take such a profile as my stochastic model to evaluate a new string, or I could generate random strings from it, where each position is independently drawn from the distribution given by that column's probabilities. And so I can write this conditional probability maybe we do a tiny example just to make clear the definition is understood. If the profile looks like, so I have A, C, G, T, then I have the columns 0, 1, 2. I'm just writing the profile as an inline table, whatever encoding P really uses. Suppose here it's, I don't know, A has 0.5, C has 0.2, 0.2, 0.1. Maybe here it's 0.2, 0.5, 0.2, 0.1. I'm only going to use the same numbers so I don't mess up the sum. Then the conditional probability of producing AAC under this profile would be the entry in the first column for A, 0.5, times the probability for A in the second column, and then the C in the third column. So it would be 0.5 times 0.2 times 0.2. That's the conditional probability of generating this thing among all possible strings of that length. Okay. And so here, what I'm doing for the next string that I pick my window from, I iterate over all the possible start positions, take the length k substring, and compute how likely is that to arise if I'm assuming the current profile from the previous rows as the truth. And then I just pick the one that's most likely. That's what this does. It's a most likely continuation. And then I'm doing this over all the rows. So I keep growing my selection of substrings. Each time I'm picking the one that, assuming the prefix I've computed so far is the truth, is most likely among the options that new string gives me. And if I've done this for all, I've selected a certain collection of substrings. Now I can check if their score is better than what I previously had. And here, this just computes the maximum or the minimum over all the start positions in the first string. Right? For each position in the first string, the rest is completely deterministic. And that also means the whole thing is deterministic. There's no randomness involved so far. Even though it uses these probabilities, it's just using them to make a selection, but I'm picking a deterministic maximum. Okay. It's deterministic, but is very sensitive to the order in which I look at things. And the order in which I list the genes, that's very arbitrary. And so it might work or it might not work, and it depends on which is my first genome mostly, or it also depends on the order of the others. But the first one I'm brute forcing over the other, I'm just doing greedy stuff, so it's a big difference. And that seems, for the example of finding this gene in plants, it seems to not work at all. There's another subtle problem, which brings me back to this picture. If you do what I described so far, it certainly doesn't work well. But the reason is stupid and fixable. In the profile as we defined it, there's a ton of zeros. If you look at this, or I mean, the counts is the same, there's a lot of zero entries here. And because we're taking products, if you have any column that contributes a 0 to your position in the string, your likelihood is 0. So especially initially, if you think about the first step, the first is you pick some substring in the first row, then you look at the second row. The profile is going to be 100% deterministic, right? There's only one character each, so the profile will say, first column has to be an A, second has to be a Z. Everything else is probability 0. So it's only going to pick, either there's an exact match in the second row, then you pick that one, or it's going to pick equally likely any of the others. That's not that useful. So in a way, we're too strict. It's going in the right direction. This maximum likelihood idea seems sensible, but we're way too strict. And the problem is that something times 0 is exactly 0 no matter what the other thing was. So we can fix that in short by starting to count at 1 instead of at 0. There's many ways to justify this. Starting at 1 is not magic. You could start at some other number. All that it does is essentially you're mixing in a prior distribution that is uniform, and then you're doing Bayesian inference on when you see characters, it moves away from the uniform, but in a smooth way. If you don't start with 1, but with some other parameter, your prior is not uniform, but some Dirichlet distribution with a different parameter. I mean, I'll leave that fun to the statisticians. For us, if you're collecting enough columns, it doesn't matter all that much what initial value you put, as long as it's not identically 0. So this is an important fix, if anything, of this sort with using profile-driven probabilities. You have to start with something other than 0 when counting. This is sometimes called Laplace rule. I think it's somewhat folklore and goes by many names. But keep that in mind whenever things seem not to work in some kind of statistical inference thing where you expect non-exact answers. Maybe start counting from 1 is already fixing it. Okay, there's this other problem that really this is so sensitive to the order, and we have no good reason to prefer one order over the other. That it's not clear how to really fix this one. There's another approach that is very general. We start with something random, and then we try to locally improve it. This one was a bit ad hoc. It started also with something, quote, random by picking any starting index. And then we try to brute force over that first choice, but then the rest was kind of greedy incremental. Maybe we can do something else instead. We start with a random choice among the collections, among the possible starting indices. And then for that collection, we try to modify it to make it slightly better. And I don't really know how to better put this. So let me just first give some pictures. And the code maybe is self-explanatory if you read it, but I don't want you to first listen. So if you take some initially random selection of these things, that's going to give you some consensus string. I called it T bar earlier. Let's use that. That's short. Now I can take that consensus string and go back into the strings and select the most likely red thing for that consensus string. So from the starting indices, I can go to some T bar. And from those, I can go to some other starting indices. And then from those, I can go back to a new T bar that might be different. And I can keep going. And in each step, I'm only going to make the score exactly better, or it stays the same. It can never go worse by definition. I'm choosing the update from S to T bar so that it exactly is the best choice for T bar, and that if I pick the optimal T bar, that gives me exactly the definition of this DH. And if I go from T bar and find a better placement of these red things, that also is only going to reduce the cost. And so in code, it looks more complicated than it really is. You start with a random initial selection here. You compute the profile. Then you pick for that profile, instead of using the consensus string, is more robust to use the profile again. You could use the consensus string, but by the same reasoning, consensus is this hard maximum. It's a bit more robust to use the profile. Then you pick the most likely starting position given the profile. If that's better, then you use that instead. And if that hasn't improved the current solution, you're actually stuck. It's not going to work if you try again. And then you just stop. That's why it's a hill climbing method. It finds a local optimum. You have no guarantee if that local optimum is any good compared to the global optimum. I guess it should climb down because we're minimizing, but ignore that. Kind of hill climbing is always formulated as a maximization. You're rolling down into the local valley. That's another option. Once you selected this random starting position, it's deterministic. And it's always going to reach some local optimum. Just no idea how good that is. In general, that method, if you just run it once, is not performing well. What you can do is run it many times and give a certain balance between how long you keep trying to make improvements. Maybe you stop it early because if you're in a very bad space, local improvements are not going to cut it, and you just stop it and run it again with a new random starting point. So you would probably want, because of that, you want to repeat a few times. Stop the hill climb early. But unfortunately, we don't have an analysis of how to choose these parameters, how often to try, for how long to try. Unlike maybe things like Schöning's 3-sat algorithm, where you could precisely analyze how long you have to run it so that it's in expectation finding the right answer. So this, in general, nothing known as far as I know. Okay, we're out of time with the planned 5-minute stop early. There's one last method. Maybe we'll do that next week then, unless you want to see the final answer from the book. It's a twist on this one. Basically, this argmax can be weakened a bit. There you go. You also start with a random initial choice. Here it's formulated to repeat a specific number of times because this is not necessarily reaching an optimum. It keeps trying. And this time we're just replacing one of the red collections. So we have our current selection of substrings. Then we're randomly selecting one of them, this one. And now we're going to get rid of the old one and choose a new one for that, but just for this single one. And that turns out to be a bit more robust. Oops, stay where you are. So we're going to update that to a new one, maybe over here. And how do you do that? You compute the profile for everything without that row. And then you could have done the same as before, take the most likely new position. A twist here with this, that's then called Gibbs sampling. You compute for every possible position in this string where we're selecting a new thing from. We're computing for each possible position the probability that it arises under the profile. And then we sample with probability proportional to that. So previously we would have said a hard max, find the most likely position, take that. Now we're saying, let's do a random choice, and the probability for picking each position is the likelihood under the current profile. So that means it can also go back to worse solutions with a certain probability. And that's why it's not always terminating, but then it also has the chance to escape local optima. And yeah, the rest is, I think, same as before. This method seems to be working best for this kind of problem in the applications where people use it for modifying. This is data dependent. But it seems this choice is more robust than taking the maximum, even if it's a bit slower. Now that's really it for today. So we've completed all the hidden messages. That's good. No more secret messages.